My name's Gerd Krause. I'm a research associate currently at the Africa Health Research Institute, so based in Durban, South Africa. I did my high school years in KwaZulu-Natal and studied at the University of KwaZulu-Natal in Pietermaritzburg. And then basically just moved down the road to start my career in immunology. And I'm still with my same supervisor, Prof. L. Leslie, at the Africa Health Research Institute. I started off doing a PhD in biochemistry. I enjoyed the lab. I enjoy working with my hands, being practical in the lab. So I have a real passion for lab work. And it started with biochemistry. My project was based on malaria though. And so from there I transitioned. I always had a passion for infectious diseases, and so moving to a TB laboratory was also of real interest to me, and to carry on with lab work. I really enjoy that. And at the Africa Health Research Institute we have access to amazing infrastructure and amazing equipment that we get to work with. And so it was sort of a day and night difference between where I did my PhD to where now I got to do my postdoc. And I really enjoyed it. We do actually have malaria in South Africa, and especially I think with climate change, the vector is a mosquito, and so the vector is actually coming down into the northern KZN areas in South Africa as well as Limpopo provinces. So we do have seasonal transmission of malaria in South Africa. There was a really good program by our government to eliminate the vector. So there was indoor residual spraying, and that pushed it back up into Mozambique. But now, I think more so that the Mozambican government hasn't been able to sustain that program. It's pushing back down into South Africa, basically. So malaria is not as big a problem as it is in most of Africa, but it's a threat to South Africa, that's for sure. I'm really passionate about the research. I think we have access to amazing cohorts at RE. So we get to work with human tissue, which not a lot of institutes have access to. And a lot of TB research internationally has access to plasma samples, for example, sputum samples, which we also do. But in addition to that, we have access to tissue samples. So we get to look in tissue at how your immune response is actually trying to counter the TB infection. So that's really interesting, and my specific interest is on a type of immune cell called B cells. These are the cells in your body that produce antibodies. That's their main sort of canonical function, but we are also interested in the alternate functions that they might have. And it's very interesting that within tissue they seem to be involved in a tissue-specific response, and that's what we are investigating. There's kind of contradicting evidence for the role of B cells in TB. So it's kind of an understudied field. So B cells aren't really highly appreciated in TB because, as you mentioned, the bug manages to hide very well from your immune system, and therefore B cells producing antibodies, those antibodies aren't very effective at neutralizing or having an effect on TB. And so for that reason, we are interested in researching the alternate roles of B cells. So B cells actually play a role in tissue in priming an immune response, and so they can actually prime cytotoxic responses through other cells. So they have these secondary functions, and that's what we're really interested in. I think in the last 3 years we played a big role in the COVID pandemic. So RE actually played a big role there, and I think we were placed strategically quite well in that sense to adapt to working on COVID. So I got to work together and collaborate with quite a few different labs in South Africa, which was really great. And I think the nice thing there was also that COVID actually brought out a collaborative spirit between scientists, which isn't often the case. A lot of times each scientist tends to find his niche of interest and to remain in that niche, whereas with COVID it sort of forced us all to work together. So in the last 3 years, we got to publish a few papers on COVID, looking at the immune response to vaccines, and trying to see if we were scared that there would be a bigger impact on the HIV positive population, so people living with HIV. And fortunately, this was not the case. But that was sort of a big scare or concern that we had. My interest being on tissue, and actually researching specifically how B cells are actually, what their impact is on the tuberculosis infection, and how they can actually help us counter the infection. So I would really like to carry on with studying the immune response in tissue, and specifically B cells. And the cancer field is actually using B cells at the moment to try and harness them to prompt these cytotoxic responses through cytotoxic T cells, for example. And I think that there are very interesting parallels between cancer and tuberculosis. And I think this is potentially technology that we could harness for tuberculosis. So I would really like to see and do sort of experimental work to shift TB potentially in this direction for either vaccines or treatments, so preventative vaccines or therapeutic vaccines, for example. SANTHE's been it's been amazing. I managed to get a SANTHE fellowship last year. So I managed to get a grant, and this allowed me to do work in the United States. So I managed to go to Colorado State University, and this was very exciting because it was a collaboration that I'd initiated through a conference that I attended in Paris, and then that collaboration actually came to fruition through SANTHE, through a SANTHE grant. So I got to physically go to this lab in Colorado State, and experience or was exposed to new technology, which we have now actually transferred to RE. And so my supervisor, Al Leslie, has actually purchased this instrument, and now we are using this instrument at RE and establishing this technology in South Africa. And it's actually the first in Africa. So it's, I think there's been, I've been very privileged to have this affiliation with SANTHE. So it's called a CytAssist instrument, and it allows us to do spatial transcriptomics, which is sort of at the, it's very interesting technology, and you get so much from, it's based on traditional microscopy, but instead of just doing traditional staining for microscopy, you can actually look at the transcripts that each cell is expressing. So in essence, the kind of recipe that each cell is transcribing out of its DNA recipe book. And then those recipes are what we are able to look at using this machine. So you can actually look at what every single cell is busy doing at that moment that it was sectioned and put onto the microscope slide. So this is very exciting, And it's been a very, very helpful course. I think a lot of us, first of all, just being put together in this group of 15, I think that's been amazing to meet other fellow early career scientists through the SANTHE program, to be exposed to their experiences, all of us being African scientists, and just to see what the challenges are, and to be able to sort of bounce ideas off each other of how you overcome certain challenges. And then to actually gain a lot of insight into possible ideas that you can implement in your own sort of mentorship programs. We did a lot of time management, assertiveness, so there's a lot of pointers that we can take from this course, and I think those will be very useful going forward, and I look forward to implementing them in my mentorship. I think we've been very privileged in the institute that RE is, in the support that RE gives its fellows, and also through the support that we get through structures like SANTHE. I am very passionate about this work, and it's just the exposure we get to other groups. We get to collaborate with a lot of different groups, international as well as national, and it's only been through the institution or the institute that RE is, and the programs such as SANTHE that are on offer. Yeah.